Okay, this is the final topic in area of study 2 for Unit 3, and it's looking specifically at immunity. So we've obviously spent a lot of time looking at the immune system and how it fights different types of pathogens, but now we're kind of looking at this specific idea of having immunity. So in this video, we're going to go through kind of 4 key dot points. One is looking at different types of immunity, looking at vaccination programs and how they provide immunity. We're going to look at some deficiencies and malfunctions of the immune system. And also, this is probably the newest thing here, is a particular type of treatment called monoclonal antibodies. So what does it mean to have immunity? I want you to think about this for a sec. You can pause this, but what does it mean when we say somebody has immunity? So immunity literally means having the antibodies for a specific antigen in your body. So for example, if you've had chickenpox before and you produce antibodies to fight off that infection, you have immunity because you have the memory B cells with the antibodies specific for the antigen on chickenpox in your body. There's a few different ways in which we can gain immunity, and they're kind of classed into 2 different kind of categories. The first is whether it is natural or artificial, and we'll look at examples in a second, but natural immunity basically does not involve any medical intervention. It just happens naturally. Artificial immunity involves medical intervention. Again, we'll look at examples in a second. The other thing that's quite relevant when we look at immunity is whether it has been gained actively or passively. So we're talking here about where those antibodies come from. Remember, something that is active requires effort, it requires energy. So if you have active immunity to something, it means that your body has produced those antibodies. Whereas passive immunity is when you receive antibodies from another organism. Okay, remember, this is receiving the antibodies themselves, not receiving the pathogen or the antigen that causes the immune response. So I guess really active is that your body has gone through an immune response. Right, so there's 4 kind of particular types of immunity, I guess. We can have it being active and natural, active artificial, passive natural, passive artificial. It'd be good if you pause for a second and think, what could be examples of this? How do we naturally create our own antibodies? So this is the obvious way of contracting the pathogen. So getting sick, and then your body goes through an immune response. You produce plasma B cells, some of them become memory B cells, and they stay in your body and they have those antibodies for future. Artificial active immunity is what happens when we get vaccinated. Okay, so you still go through this active process of producing B cells and producing antibodies, but it's artificial. There was some medical intervention there. Okay, so that was through vaccination. Just I guess an interesting differentiation here. People often ask, what's the difference between vaccination and immunization? A vaccination is this. It's actually getting a vaccine. Immunization is when your body has gone through that immune response and you are now immune to it. So there's usually a 2-week window between being vaccinated and then actually being immune to something, because that response takes time. Okay, passive natural immunity. So remember, this is antibodies coming from a different organism, occurs through the placenta or through breast milk. Okay, so this is how infants, and not just humans, all different types of animals, receive immune protection from their mother, either through the placenta while in utero or through breast milk. And this is really, really important for those early months when an infant doesn't have any immunity, okay, and often before they're able to get their vaccinations because they're too young. And then the last category is passive artificial immunity. So this is when you receive antibodies that another organism has made through medical intervention. And an example of this is antivenom. Antivenom is really interesting if you want to look it up, but you don't have to go into too much detail. But basically, antibodies, antivenom is made by injecting an animal, usually a horse, with the venom of, say, a particular type of snake. Let's say it's a brown snake. That horse will go through an immune response. It will produce antibodies. Those antibodies will be extracted, and then they'll be put into a serum. And then if a person presents to a hospital after having been bitten by a brown snake, they can inject them with these specific antibodies. These are crucial for things like toxins that are so potent that they would kill a person, before there's an opportunity for them to go through a natural response. Okay, so generally, like I said, these are for venoms and toxins. So looking at vaccines in a little bit more detail. So vaccines elicit an active immune response by exposing a person to an antigen. Okay, so remember, it's the antigen that during an immune response gets presented on an antigen-presenting cell and travels to the lymph nodes and activates a helper T cell. Okay, the antigen is actually the bit that matters. Obviously, I think there's a lot of misunderstanding about vaccines, and a lot of people think, oh, well, you're just injecting a person with a disease and that's really dangerous, it's going to cause the disease. It's not quite how it works. If we look here at this virus, okay, this is, a full virus. It's got the genetic material in here, either the DNA or the RNA. It's got its capsid, and then it's got all these antigens. There's a few different types of vaccines. They're very complex in terms of how they're made, and certain types work better for certain pathogens. One type is having a live attenuated vaccine, which basically means a weakened version of that pathogen. So if we look at this picture here, it's quite hard to see, but the genetic material has kind of been broken a little bit. So it's not a fully functioning vaccine, but what does it have? The antigens. Sorry, a fully functioning pathogen. It has the antigens, and that is enough to cause an immune response. We can also have inactivated or dead pathogens where the genetic material has been removed completely. So that virus has no way of replicating within the body, but again, it is able to elicit an immune response. There are some vaccines that are called subunit vaccines, and that is literally just the antigens. Okay, it doesn't have any of the other material. And then the final type, and you might have heard about these in the news, are gene vaccines. And the Pfizer mRNA vaccine that is currently being used for coronavirus is an example of this, where instead of even injecting the antigen, they inject the RNA sequence that will make that antigen. Okay, so it's still enough. Your body will then produce the antigen to produce an immune response for that particular pathogen. Okay, so they're just some examples of how vaccines can be made. And this diagram just it's really just a recap, I guess, of the immune system. The only thing that's different is this part here. Okay, so let's say this is the skin. You get injected with whichever type of those vaccines it is. So let's say it's this, let's say it's a live attenuated vaccine. Your body will go through its local inflammatory response here. We'll get macrophages and dendritic cells will go to the area. Phagocytosis will happen. Remember, these are antigen presenting cells. Okay, so they're going to travel to the lymph node with a bit of the antigen on their surface. They will present to a helper T cell, and that helper T cell is going to activate a humoral response. Remember, a humoral response happens whether it's a viral or a bacterial infection, and then, of course, we get our full immune response. Okay, so this diagram, I guess, is a good summary of the other things we've looked at, but just changing the mode of the antigen entering the body. In the last video, we talked about primary and secondary responses, immune responses, and booster shots are a really good example of this. So some vaccines only create a very small primary primary response. So you can see here, we're looking at the antibody numbers. We get some antibodies made, but then after time, it actually really drops down, and it's not enough to provide an immune response. So some vaccines require a booster shot. Remember, at the time of this booster shot, the memory B cells exist already. So when that booster shot with the antigen is given to the person, the body responds much quicker, we get a much faster response, and we get higher numbers of antibodies. Some vaccines in your life you will have, every 10 years. They're an example of this, where the antibodies don't remain in the body for long enough, so booster shots are required. All right, just quickly going to talk about herd immunity. And again, you've probably heard of this in the news in recent times. Herd immunity refers to the indirect protection that occurs when a certain percent of the population is immune to something. And generally, this is through mass vaccination. Technically, herd immunity could occur if 95% of people in an area contracted a disease and developed immunity to it. It'd be the same thing, but we're basically talking about having enough people immune that an infectious disease can't spread. So if you look at this picture here, we're looking at, this is when there's no immunization. So we start here, the red people are contagious. They've got an infectious disease, and then all the blue people are susceptible, meaning they are not immune to this disease. Okay? Obviously, the nature of infectious diseases, certainly some of them, is that if we allow enough time, we end up with this. And I think, coronavirus is an example we can look at. Obviously, in Australia, we've had other ways of protecting from infection, but in a lot of countries, just in less than a year, huge amounts of people have been infected because no one had immunity to this disease, or not many people had immunity to this disease. There would have been some natural mutations that allowed immunity. Okay, so over time, we've got almost everyone infected because there's just these viable hosts all over the place. Over here, this is with immunization. So now we've got the red are infectious, the yellow are immunized, so they are immune, and then the blue are susceptible. So with this, we can see that if all the people around this person with infection here are immune, then the virus or the pathogen can't spread. It's got nowhere to go. It needs a host, and it is not going to reach susceptible people as easily. So, of course, you'll still get some people might get it, but in general, it protects susceptible people. What we're talking about here generally is people who can't be vaccinated. Okay, so as I mentioned before, babies can't be vaccinated for a few months. A lot of elderly people can't have certain vaccinations, or people who are immunocompromised, meaning they have weaker immune systems, and we'll look at this later, but their immune systems are so weak that even introducing an antigen through vaccination could be fatal for them. Okay, and just a fun fact that the general recommended percentage of vaccination to have herd immunity is around 95%. Okay, so we've looked at the different types of immunity and the vaccination programs. We're actually going to jump down to the bottom dot point here because I think it flows a bit better from what we've just been doing. So looking at monoclonal antibodies. Just quickly pause for a second and try and recall what is it that causes cancer? At a scientific level, what is happening that causes cancer? Okay, so cancer is the uncontrolled division of damaged cells. Okay, so remember, normally if a cell is damaged, it will receive a message to go through apoptosis and it will leave our body and won't cause any problems. Cancer happens when those signals to destroy aren't received properly and those damaged cells keep dividing. Like I said, normally they'll be recognized by the body's own T cells and destroyed, but this doesn't happen and we end up with tumors. This is important for what we're going to look at next. So monoclonal antibodies are a type of treatment, and it involves producing antibodies in a lab, and they're called a monoclone because they're basically a clone of a single type of antibody, meaning that all of the antibodies are identical to each other, and they're identical and specific to certain markers found on cancer cells. They can be used to treat a variety of autoimmune diseases, but particularly we look at them in relation to cancer. But let's say we'll look at how they are made and how they work in a second, but this is a monoclonal antibody. Let's say this is a cancerous cell and it's got a particular marker on it. These monoclonal antibodies will be made, produced to match with the marker on that cell, and then that's going to flag that cell to the immune response, and maybe, you know, some cytotoxic T cells are going to come along. They now see this antibody and they will encourage those cells to destruct. All right, the steps of making these antibodies, there's an image that goes with this. There's quite a few steps, so I'm just going to have to put them on this slide, but I do encourage you to have a look at the image as well. So this is how they are produced in a lab. Step one is that a mouse is injected with the target antigen. Okay, and remember that antigen is from the type of cancer cell or the type of cell that we want to target. If you think for a second, what will happen when a mouse is injected with an antigen? It will go through an immune response. Okay, so the mouse will react to that antigen. It will produce plasma B cells with specific antibodies. Okay, so just like a normal immune response. Those plasma B cells are then extracted from the mouse. Okay, so those specific plasma B cells that the mouse has produced in response to this original antigen are extracted, and then they are mixed in a dish in a lab with a specific type of cell called myeloma cells. So we've basically got the B cells from the mouse that have the antibodies that we want, and then we have these myeloma cells. Just an important thing to note, when it says here that they're cancer cells, they're nothing to do with the type of cancer that we're trying to treat. They're just a specific cell. They will fuse together, the B cell and the myeloma, forming a hybrid or a hybridoma. The reason for these myeloma cells, if you remember, myeloma cells divide. Cancer cells divide uncontrollably. So that's actually good, because what we want to do is clone this cell. We want these antibodies. So when you mix the two of them together, we're getting the antibodies from the B cell, and also we're getting the division properties of the myeloma cell. So we basically get these cells that continue dividing and make heaps and heaps and heaps of clones of these antibodies that we want. As I said, they're collected and grown to produce those antibodies, and then eventually the cloned antibodies, which are specific to this antigen that was injected into the mouse at the first time, are extracted, and then they are injected into the patient. So this is that diagram I was talking about. So the antigen is injected into the mouse. The mouse goes through an immune response. It produces these B cells with antibodies all over them. Okay, the antibodies and the B cells and the myeloma are mixed together. We get these hybridoma. Again, just imagine that these hybridoma are covered in those antibodies that we want, and then the antibodies are extracted. What happens next is that those antibodies obviously will find those target cells. So here we've got a cancer cell. The antibodies injected into the patient will kind of bind to those cells. And then there's a few different ways that they can be used to treat cancer. One is that they will just attach, like I mentioned before, they attach to the cell and they kind of make it more visible to the immune system. So maybe our cytotoxic T cells will come along now, and destroy those cells. Another thing they do is that they block certain receptors. So let's say this receptor is the receptor for growth signals. Okay? If an antibody is blocking those receptors, that cell cannot receive the message that it needs to grow, so they won't grow. Basically stops these cells growing. Another thing they do is they might block molecules that attract new blood vessels to the area. So remember, obviously blood carries oxygen, which is needed, for cellular respiration for anything to grow. If we block blood from coming to that area, more cancerous cells are not able to grow. And then one kind of really cool, quite futuristic way of treating cancer with monoclonal antibodies is combining them with particular radioactive particles. They might have these particles on them, as well as chemotherapy drugs, meaning that the drugs can be delivered directly to the cancer cells. One really big downside of chemotherapy is that it just attacks all cells, which is why people get really, really sick when they go through chemotherapy treatment. It can't tell the difference between cancer cells and healthy cells. So if we could target it just to the cancer cells and leave healthy cells alone, obviously it's much, much better for patients. All right, the last thing we're going to look at now is looking at deficiencies and malfunctions of the immune system. And the study design specifies multiple sclerosis, HIV, and allergic reactions. We can classify these diseases into three kind of broad categories. First, we have autoimmune diseases. Okay, auto means self. So this occurs when a person's immune system attacks its own cells. Okay, and you've probably heard of different examples of this. Sorry, they should not be there. Autoimmune diseases are when the body accidentally attacks itself. And, it can be very, very severe and cause a lot of problems. Like I said, we're going to look at multiple sclerosis for this. There's also immune deficiency, or immunodeficiency, it's sometimes called. So this occurs when the immune system is not functioning properly. So usually something is attacking the immune system. As a result, a person can't fight infections properly. So if somebody has immune deficiency and they get a common cold, it can make them really, really, really sick because their body can't fight that normally quite simple infection. These can occur congenitally, so a person could be born with an immune deficiency, or they can be secondary to other things. And AIDS is an example of that, which we're going to look at in a minute. And then allergic reactions. These occur when the immune system overreacts, okay? And it's usually to non-pathogenic substances. So what we mean by this are things that aren't pathogens, that aren't going to cause us to be sick, but for some reason the body responds to them as if they are pathogens. I'm sure many of you have experienced this in the past, but things like dust and pollen are really common examples of, pollen on its own doesn't actually make people sick, but people with allergic reactions, their body responds to pollen as if it's a pathogen. So we'll look at multiple sclerosis first. Like I said, this is an autoimmune disease. It is when the immune system attacks neurons or parts of the nervous system. This happens when T lymphocytes, so T cells, don't recognize the self-antigens on these specific cells that are called myelin-producing cells. Okay, so this is a neuron, okay, and we've got these cells that produce the myelin sheath. Now, the myelin sheath is really, really, really important in transmitting nerve messages. It makes the nervous system run really quickly. So remember, self-antigens, the job of a self-antigen is to tell the immune system to not attack. But in this example, the immune system doesn't recognize them. So what does it do? it attacks. It attacks these cells that it shouldn't. And you can see here, we've got these T cells that haven't recognized the self-antigens. They launch an immune response. We get plasma cells activated. They release antibodies that, attack the cells. We get macrophages coming along and destroying those cells. If we were to zoom into a myelin sheath here, this is healthy myelin. That's what it should look like. Provides an insulating layer around the nerve fiber. This is a nerve affected by MS. So that myelin sheath has been destroyed basically by macrophages and things because the body didn't recognize it as being self. As you can imagine, if you get enough damage to nerve cells, the symptoms are going to include things like progressive weakness. So over time, a person will get really, really, really weak, often to the point of not being able to move. A lot of pain or altered sensations, and poorly controlled movements. So someone might be able to still move, but very, very slowly. The next example is looking at immune deficiency, and particularly looking at HIV. Okay? HIV stands for Human Immunodeficiency Virus, which kind of makes sense now when we look at what actually happens. Really important to note here, HIV is the virus, okay? It's a pathogen. People often get quite confused between HIV and AIDS, and hopefully this will kind of clear that up a bit. This is what HIV looks like. It's a specific virus. It's transmitted through blood or through mucus contact. So mostly it is through either sexual contact, through sharing a blood product, so that might be through blood transfusions or sharing syringes, and it can also be passed from a mother to a baby in the uterus or through breast milk. The virus, remember all viruses need a host, okay? And different viruses like different hosts. The HIV virus uses a particular type of T helper cell, so CD4 cells, as their host, okay? So this virus targets cells of the immune system, helper T cells. Over time, those CD4 cells just are under attack by the HIV virus. And when I say over time, this can be over decades. It can take a really long time before someone is even aware that they have this, because very slowly and very quietly, their helper T cell numbers are just decreasing, and their immune system is getting weaker and weaker. If this is left untreated, it's important to know at the moment there is no cure for HIV, but it can be treated. Basically, we can stop this happening. We can stop it spreading within a person's body and stop their helper T cells depleting. But if it's left untreated, this leads to AIDS. And I've got a graph on the next slide to show you. But AIDS is a disease or a syndrome, I guess, that is caused by HIV. It stands for acquired, meaning that you weren't born with it. It was acquired from something. Immunodeficiency syndrome. Okay, so really important just in terms of a lot of misunderstanding around HIV and AIDS. People don't catch AIDS, they catch HIV, and then over years they can develop AIDS if their immune system becomes weak enough. Generally, this is the point at which the immune system is so deficient that it can't fight off other infections. So people, that die from AIDS-related illnesses might die from something like pneumonia because their body can't fight it off. This graph just kind of shows what happens over time. So we're looking here at the CD4 count. Remember, CD4 is a helper T cell. Okay, person gets infected over here, acute infection. There's an interesting thing where initially after that they get a depletion in CD4 cells. So people might often get some symptoms here. They might get a bit fluey or have some symptoms. Then the CD4 numbers actually go up a little bit, and then over time it just really, really drops. There's no set time. It's not a thing that after 10 years someone has AIDS. It really depends on how long it takes for these CD4 cells to deplete. So you can see up here, there's a really, this can be up to, 10, 15, 20 years where a person has no symptoms. Then they might start getting symptoms, and then AIDS is technically when it's below this line, when their CD4 count is less than 200 cells per cubic millimeter. You do not need to know that, but that's the clinical definition of AIDS. At this point, there are so few CD4 cells, those helper T cells, that if you think about what helper T cells do, we can't stimulate a cell-mediated response, because remember, helper T cells are what stimulate cytotoxic T cells to divide, meaning a person can't fight off things like viral infections when they have infected body cells, but also helper T cells activate clonal selection of B cells, which is our humoral response. Okay, so at this point, the person's helper T cell numbers are so low that they can't fight off infections, and unfortunately that quite often does lead to AIDS-related deaths. Okay, almost there. The final topic or section in terms of immune diseases is looking at allergic reactions. Like I said, this is when things called allergens, which is basically non-pathogenic agents and not pathogens, are recognized by the immune system. You do need to know the steps of this. It comes up quite often in the exam. This says an allergen. Let's just assume this is pollen, okay? Because that's what the study design says. So pollen, which is found in plants, and during spring it's blowing around all the time, and people often get allergic reactions. So the steps of this. There's always an event known as the first exposure. Often a person doesn't develop any symptoms the first time they're exposed to an allergen. It's not really understood why certain people get allergies and others don't. Okay, so it's an area of science that if you want to go and study, absolutely do, because there's lots of unknowns. Anyway, allergen, which is going to be pollen, for example, attaches to, remember we looked at there's 5 different types of antibodies. One of those, the IgE antibody, is involved in allergic reactions. So an allergen attaches to the IgE antibodies found on B cells. Okay? Those antibodies get released by the B cell. Remember, B cells release antibodies, to go and fight infections. The difference is this isn't going to fight any infection. These antibodies get released into the bloodstream, and then they find mast cells. Remember, mast cells are your own white blood cells that are found all over the body. These antibodies bind to mast cells. Okay, so we've got these IgE antibodies, and we call this cross-linking. Okay, so cross-linking occurs when IgE antibodies specific to an allergen bind with a person's mast cells. So that's all part of that first exposure. What happens then in future, when a person encounters pollen, for example, the allergen will bind with the IgE antibodies on the mast cells, and inside the mast cells we've got these little granules, they're almost like vesicles, that are full of histamine. And hopefully you're thinking back like, histamine, that's involved in an inflammatory response. Correct, that's basically what happens here. The allergen, or the pollen, will bind with the antibodies on the mast cells, and this will cause the mast cells to degranulate, which basically means to open up these granules and release histamine. Now, if you think back for a second as to what histamine does in an inflammatory response, its job is to open up the blood vessels. Remember, it increases the vasodilation. It makes the blood vessels leaky so that white blood cells can get out and fight off infection. The difference is there's no infection here. So what happens is histamine causes the blood vessels to become all leaky, and basically causes a local inflammatory response. So if you've had an allergy to pollen in the past, you'll probably notice that your nose gets really red, it gets inflamed, it starts getting really runny. That's all the result of histamine. Okay? It's causing vasodilation and leaky blood vessels, which causes all of those symptoms, which is why the treatment for allergies are called antihistamines. They basically block the action of those histamines, because these allergic reactions are not beneficial. They're not actually protecting us from anything, because pollen itself actually doesn't cause any disease. So hopefully you found that interesting. Some of you might be able to relate that back to your own experiences. All right, I know there was a lot in that, but that is that final topic. And that is basically everything for Area of Study 2 of Unit 3. Thanks.